Hello and welcome to another video in the lecture series entitled Advanced Biomaterials. So in the previous videos or in the previous lectures we have seen about scaffolds, their fabrication techniques, the materials which we can use to synthesize such scaffolds, their applications and also some of their important characteristics which needs to be considered while fabricating a specific scaffold for a particular application. So in today's video we'll be looking at a very particular application of a scaffold, which is a scaffold for skin tissue re-engineering or skin tissue remodeling. So a scaffold to be acting as a skin regeneration template, what are the different considerations, what are the different criteria while fabricating such scaffolds, what are the biomaterials which can be used for the fabrication of such scaffolds, and all the related aspects we'll be looking at in today's or in this video. So let's start by discussing an example of a scaffold for which can act as a skin regeneration template. So a scaffold acting as a skin regeneration template. So as we know, the human skin is considered as the largest organ in the body because the skin is responsible for so many different functions of our body, and it is composed of different tissue types as well and different cell types as well, which integrate and perform different types of functions in the body. So the skin is primarily composed of two layers, the epidermis and the dermis. So the epidermis, which is the outermost layer of the skin cell, which is composed of squamous epithelial cells, the keratinocytes, and several other types of cell types as well. And finally the dermis, which is deep-seated and occupies a large volume of the skin, which is composed of mainly fibroblasts and other specific cell types. So both the epidermis and the dermis collectively make up what is known as the human skin. So in different cases, our skin is subjected to different wounding conditions. So some cases the wound may be very superficial or very simple in nature, and which can easily heal over time through the formation of a scar tissue. Or in some cases it is also seen that the extent of damage to the human skin can be in such a great extent, or can be such large amounts or large quantity that automatic regeneration might not be that soon, might not be possible, or it might lead to several other complications and other infections. Thereby external intervention might be required in order to correct such types of defects or such large scale damage to the human skin. So an example of such large extent of skin damage can be third degree burns. So these third degree burns can be the examples of scenarios where large portion of the human skin can be damaged. That is, the large portion which corresponds to the dermal layer of the skin can be damaged because of these third degree burns. So often in these extent of these third degree burns, what people do, they use meshed autograft. So autograft means using a part of the undamaged skin from a patient's own body, and replacing that part of the burned skin using that part of the healthy part of the skin from a different source. But in many cases it is observed that for using these autografts for treating third degree patients with third degree burns, a large percentage of the total skin area is actually lost. Therefore, to use a healthy tissue or a healthy skin sample from an unaffected area will imply loss of skin from that area as well, and insufficient amount of skin material to serve as a replenishment to the damaged part of the skin. So that is why external or synthetic materials or even natural materials from external sources can be used to fabricate scaffolds which can act as skin regeneration templates. So we will be discussing such a construct in today's video. So In order to treat patients with third degree burns, a skin regeneration template is used which is porous in nature and constructed using two different types of polymers. One is collagen type 1 and chondroitin 6 sulfate. So these two polymers can be used to fabricate a porous scaffold or a porous skin regeneration template with a wide range of pore size and degradation rates. So, a composite polymer system or a combination of two different polymeric systems can be used to make or fabricate a porous scaffold which can serve as a skin regeneration template which has different pore sizes based on the type of the polymer or the type of construct which is being fabricated and the different degradation rates. So usually in order to simulate or to mimic the skin, it is synthesized in two layers. The outer layer will be acting as the epidermal regeneration template and there will be a lower layer as well which will serve as the dermal regeneration template. So the pore sizes of the epidermal and the dermal regeneration templates will be different based on the type of cell type which is used to migrate in these scaffolds and the construct which is being synthesized. So the top layer will be the epidermal and the bottom layer will be the dermal template. Now this is an example of a classic wound healing model which is studied for its ability to delay wound contracture and promote the dermal regeneration. So it is used for studying the delay in wound contracture and for promoting dermal repair. So it has been found that the optimum, since these are porous scaffolds, so it has been found that the optimum pore size for studying the delay in wound contracture has been found to be 10 to 120 micrometer of pore size. In order to facilitate the migration of fibroblasts inside these cells and inside the interstitial spaces or the pores of the scaffolds and to thereby enable it to form or assume a tissue-like morphology. Right? So this has been found to correlate with a half-life or T1/2 half-life of 27 days compared to 7 days. Now this delay in wound contracture of 27 days when compared to 7 days, it has been found that it is believed that this delay in wound contracture is related to improved healing and it correlates with fibroblast migration through the template. Now there are different considerations while fabricating such types of scaffolds. So, the first condition or the first consideration is the effect of pore size on the migration of the cells. So, the effect of pore size on the migration of the cells. So as we had discussed, these scaffolds are highly porous structures, and these scaffolds can often be seeded with cells before implanting them in the damaged area. So that's once these cells are implanted in the or seeded in the scaffolds, they will start migrating inside the pores of the scaffolds. So often these pores are interconnected. So these cells will migrate deep into the three-dimensional spaces or in the interior spaces of the scaffolds, and over time they will assume a tissue-like morphology. So once these cells are assuming a tissue-like morphology, they can start performing the different roles of the tissues, such as secretion and also exerting mechanical forces on the scaffolds, thereby and also secreting extracellular matrix in the secretory cells, such as and also secretory proteins as well, in order to perform all the functions which are actually performed or which are actually seen in the native tissues. So these pore size will affect the rate of migration of these cells inside the scaffold. So an optimum pore size has been found to be required for promoting or the proper migration of these cells. So the primary cell type in these types of scaffolds are the fibroblasts, and these pore sizes correspond to the optimum migration of the fibroblasts in the scaffolds. So what is the significance of these pore sizes? So if the pore sizes are very small, then these fibroblasts will not be able to migrate in the interstitial spaces of the scaffolds or in the pores of the scaffolds. Thereby, the whole purpose of the scaffold will be diminished. And if the pore sizes are very large, then the surface area which is available for the cell's attachment will decrease or will decline. So the cells will not be able to attach to the scaffold with the desired contact area which is required for it to assume the tissue-like morphology. Then there is another important consideration for fabricating such skin regeneration templates, which is the degradation rate of the template. So the second criteria is the degradation of the template. Now, based on the purpose for which the scaffold is used, the degradation rate of the scaffold will play a very significant or a very crucial role in determining the fidelity of the construct. That is to say that if the tissue which is needed to be replaced has a very slow generation rate, then the degradation rate of the corresponding scaffold should also be in par or at par with the degradation or with the generation rate of the cells. For example, if we are trying to make a scaffold for promoting repair of bone cells, then the degradation rate of the scaffold should be slower because the rate of genesis of osteoblast is comparatively slower than the generation rate of fibroblasts. So, based on the generation rate of new cells, the degradation rate should be directly correlated with it. So if the generation rate is high, the degradation rate should be also be high, and if the generation rate is low, then the degradation rate should also be low. So if we take the example of this skin regeneration template, then the degradation rate will be found to be optimal when tuned to appropriate normal wound healing rate. So there will be a correlation between the degradation rate of the scaffold and the rate of normal wound healing. Or tissue regeneration. So there will be a relation between the degradation rate of the scaffold and the normal rate of wound healing. So if the rate of degradation of the scaffold or the template is considered as TD, which is the degradation rate of the scaffold or template, and the rate of normal wound contracture, normal wound healing, if it is TS, then the relationship should be such that TD by TS should approximately be equal to 1. Right? So this is a very important relationship to be kept in mind while considering a skin regeneration template for the construction of a skin regeneration template, or any scaffold for construction or for repair of any particular tissue type. That is, the degradation rate of the scaffold should be comparable to the normal rate of wound healing, so that the tissue which is being formed is actually, and the material which is degraded, they should be equal. So if the tissue formation is faster than the rate of template degradation, then the template will persist and will actually inhibit new tissue formation. And also, if the rate of degradation is very fast compared to the rate of normal wound healing or tissue regeneration, then the fresh tissue which is growing, or the cells which are growing, will not have that mechanical support to grow and to proliferate. So the effectivity or the fidelity of the construct will depend on the relationship between the degradation rate of the scaffold and the rate of normal wound healing. So it has been TS, the rate of normal wound healing in case of these 3rd-degree burns, it is found to be 3 weeks for skin, whereas the degradation rate for this specific scaffold which we have discussed, it was found to be 2 weeks. Therefore, this relationship will actually hold, and therefore the specificity or the fidelity of the construct will be maintained. So in today's video, we have seen that how we can actually fabricate a 3D porous scaffold for which can act as a skin regeneration template. We have seen that what are the different factors or the different parameters which can be which need to be kept into consideration, kept into mind while fabricating these types of scaffolds. We have also seen the 2 important considerations, that is the effect of pore size of the template on the migration of cells, and also the rate of degradation of the template, how it can be directly be used to modulate the different physiological properties of the skin regeneration template. So with this we'll end today's video, and in the subsequent videos we'll look at some other types of templates and some other types of important biomaterials or polymeric systems which can be used for constructing many other interesting constructs which can be used in different applications in tissue engineering and other fields of biomedical engineering. So with this we'll end today's video. Hope you enjoyed it. Thank you. See you in the next video.